what I tried to do in this talk is really start out with a very broad perspective, and in fact, I have to make a confession at the beginning of the talk that I am a microbial chauvinist, and so I'm really focused on why microbes are important in our ecology today, but also over the course of the history of our planet. So I'm going to give a general introduction. I'll talk about some of the challenges of how to study those organisms we can't even see, and then get into some of the science that we've been doing more recently. So that will be the course of our talk today, and we'll see how it goes. So first of all, I just want to tell you a little bit about why microbes in general, bacteria, archaea, protists, are so important to the functioning of our planet today, and also in history. So the oceans formed, we think, about 4 billion years ago, and not too long after that, life came on the scene, maybe 3.8 billion years ago, in the form of very simple microorganisms without complex organelles and that sort of thing, the prokaryotes, bacteria and archaea. Not very long after that, photo systems evolved, biochemical systems that can capture light energy. And not too long after that, cyanobacteria that can take sunlight energy and split water and make organic carbon out of CO2 evolved. And of course, that led to the evolution of oxygen over time that eventually set the stage for the evolution of eukaryotes, multicellular eukaryotes, and the ones that we're familiar with today. And so the point of this slide really is that those microbial activities and evolution over time set the stage for the sorts of life forms that we're well familiar with now on Earth today. So that's one important aspect. This slide is way too busy, and I almost hesitated to use it as a second slide, but there's a very simple point to make here. What you're looking at in this blue area here is biotic reactions that occur on life, and the issue really is that energy for living systems really has to do with electron transfer. They're oxidation and reduction reactions. And that's the really currency of energy in life. And so all these transformations that you see here are just the swapping of electrons from one form that's energy-rich to another form that can be easily reduced. So why am I showing you this? Well, I want to show you what sorts of redox reactions are the main ones for higher life forms, multicellular organisms. And of course, I just mentioned photosynthesis, oxygenic photosynthesis that evolved first in cyanobacteria, and then it basically was transferred to plants. And so this is a super important process today, of course, because this is where all primary production comes from. It's the utilization of sunlight to reduce CO2 with electrons from water to produce organic carbon and oxygen. And of course, that forms the primary food sources that organisms like ourselves can eat, this organic carbon here, so that aerobic respiration, which is what we do, and most heterotrophs do, is the oxidation of that reduced form of carbon from photosynthesis.
What's a heterotroph?
A heterotroph eats organic carbon. And so that's what this is right here, organic carbon. And they're going to oxidize that, reduce this oxygen, that's respiration, that's what we do, and that produces CO2 and H2O again, which cycles back. And the main reason I wanted to show this was just to illustrate the diversity of redox reactions that occur in bacteria and archaea. So for plants and animals and fungi, mostly this is what's going on, either oxygenic photosynthesis or aerobic respiration. But all these other elements can either be used as energy sources or as electron acceptors, essentially be breathed, in all these other different reactions that actually turn the geochemical cycles on our planet. That is, make reduced forms and oxidized forms that mobilize the elements and really cause the biogeochemical cycles to turn. So those 3.8 billion years of evolutionary history in bacteria have led to the ability to perform all these functions, and that's what really regulates the homeostasis of biogeochemical cycles in our Earth today. Let me give you a much simpler example, and this is the nitrogen cycle, which is very important in the ocean. And the reason is that nitrogen is a very important element, either in the form of nitrate or ammonia, to allow plant growth. So plants, of course, as we know, they need CO2, they need sunlight, they need water. They also need phosphorus and nitrogen in order to build new plant cells. So in the ocean, the nitrogen cycle is, again, this electron swapping game that I talked to you about before, the either stripping of electrons from one substrate and then putting them on another substrate. And that's what cycles nitrogen in these various oxidized and reduced forms throughout the ocean. They're very important processes for both generating the nutrients that plants can eat. Now, what would happen if the microbes weren't there in the ocean? Well, each one of these individual steps in the pathway of the nitrogen cycle is typically regulated by bacteria. So without the microorganisms there in the ocean, the nitrogen would basically end up in one single pool, and cycling would cease to occur. So I'm just sort of trying to drive that point home. Microbes are also found pretty much everywhere. They've had 3.8 billion years to figure it out. I think the temperature limit for microbes right now is around 114 degrees Celsius under high pressure near hydrothermal vents. They can also grow in very cold areas and even in very deep sediments in the bottom of the ocean. So they have all these different biochemical abilities, and they've also permeated almost every habitat that you could think of on Earth that has energy and water and the appropriate elements. From the perspective of a microbiologist, one of the more important things is that the forests of the sea are microbial. So unlike terrestrial systems with macroscopic plants and so on, in the sea we have very small microorganisms which are responsible for all that photosynthesis and produce about half of the annual production of oxygen each year on our planet. So these little organisms, although they're very tiny, are super important for these processes of primary productivity in the ocean and generation of the oxygen that we breathe. This is an epifluorescence micrograph. These are just microbes that are pulled down on filter and stained with a stain that binds to DNA. And if you excite that with light, the organisms fluoresce. And that's how oceanographers can count microbes in the sea. They can also use other techniques like flow cytometry. The point here really is that microbes are really abundant, so that for every teaspoon of seawater there's 1 million microbes, and there's about 10 million more viruses in each small bit of seawater. So they're super abundant. And it's not just that they're abundant, it really has to do with their activity and turnover. So estimates are that the turnover time for photoautotrophs in surface waters are about 1.5 per day. And if you take a look at that, that's basically a turnover of close to 10 to the 30th cells per year, which is a phenomenal number and really hard to understand. So that's how many cells are being produced and consumed over the course of the year. And that's a lot of biology happening in a pretty short period of time. So just to pull out all that together in perspective, I guess one of the ways that I think of microorganisms is that they're massively parallel, broadly distributed processors of matter and energy that actually cycle a lot of the elements on Earth. And here we're looking at chlorophyll of those tiny microscopic plants that are growing in the ocean. And we can see their variability as they bloom and bust in response to the supply of nutrients that's driven by hydrodynamics in the ocean. So that's sort of a very quick global perspective of why microorganisms have been important in the history of our planet, but also on our planet today. So there are some challenges, though, to studying these microbes because they're so small, they turn over so quickly in the ocean, and we can't really observe them directly very easily. So there's been a lot of advances recently in terms of how we can begin to see these unseen microbes using molecular biological techniques. And so I'm going to walk us through that just very quickly so we can see how people are using very reductionist techniques like molecular biology to do ecology in the ocean. So first of all, a little background. Before about 1980, we weren't even doing this sort of epifluorescence microscopy counting. So we didn't know the numbers of cells out in the wild. They were sort of invisible to us, in other words. most of them, and this is still true, really weren't easy to culture, so we couldn't use classical microbiological techniques to study them. And most of them we couldn't identify in a taxonomic sense. So it was really this sort of mysterious biological world of small organisms who remained largely unknown. So how did we get around that? Well, in about the 70s, a solution emerged. This fellow here, Carl Woese, had the realization that if you looked at very conserved macromolecules that are found in all living cells, that you could begin to infer evolutionary relationships by sequencing those macromolecules, in this case ribosomal RNAs, and from the differences in their sequences infer evolutionary distances and create a tree, an evolutionary tree like this one, that could interrelate using phylogenetic techniques the relationships between all cellular life forms on Earth. Using that approach, Carl was able to discover a new group of microorganisms called Archaea, and we could begin to think about relationships amongst very mysterious organisms like the protists that are found here. Here we are, Homo sapiens, relative to some of these other groups like plants and fungi and so on. So these techniques could allow us to interrelate all life forms very distantly, but also ones that might be very closely related. So how did this technique allow us to start to understand microbes in the wild? Well, that was really an extension of Woese's approach using a different twist, and that was really first proposed by Norm Pace in the mid-1980s. And the idea is this: you could reach into a mixed natural population of microbes, say by filtering them on a filter, and then you could extract the total nucleic acids, the DNA, from all those cells together. But using molecular biology techniques, you could clonally separate those informative macromolecules like ribosomal RNA genes. You could sequence those, and then you could actually put the RNAs that were present in this mixed natural population on a phylogenetic tree like this to try and understand who was present in this mixed natural population. And as an extension of that technique, you could also begin to get quantitative information on the distribution of even uncultivated microorganisms present in mixed natural populations. So for microbiologists at this time, this was a huge advance, because now we could reach into natural populations and start to understand who's there, at least from a phylogenetic standpoint. And just very briefly, what we learned, we had maybe 12 major divisions or phyla that were known in the bacteria in 1987. And of course, it's well beyond this now in 2017, but it was up around 100 major phyla of bacteria that were discovered using these techniques. There's a slight problem here, though, because all you're getting is placement on a phylogenetic tree, but you're not really learning about the physiology of these organisms, their biochemistry, their genetics, and so it can only take you so far. And so that's where some modern advances in genomics come in, because now rather than just looking at a single gene, a phylogenetically informative macromolecule, now we can look at the collection of genomes or genes present in a mixed natural population by extracting DNA from the sample, sequencing the genes or sequencing elements of those and assembling them to get to genomes or genome drafts, genes, and knowing what functions are associated with those genes, as well as phylogenetic placement of the microorganisms present in these mixed natural populations. So in a way, this approach is just a simple logical extension of Norm Pace's original idea, but now rather than just looking at a single gene, we can look at the collective genes and genomes of microbes in a population. So what I'm going to do is just tell you really a couple vignettes of how this has advanced our understanding and a little bit about where the field's headed today in the context of the ocean. So one of my first forays into this, Marion actually mentioned, and that was looking at planktonic bacterial communities, and it was one of our first attempts at doing this. And what we did was to sequence large fragments of DNA from this mixed population of microorganisms in the ocean. And one of the first surprises we saw when we annotated these genes, these little colored bars just represent individual genes of different functional types, and they're colored by what particular pathway they might be in. But this gene here really surprised us because it was an opsin, which had never been seen in bacteria before, so that was surprising. But this is also a little bit cautionary, too. Just because you see a gene that resembles another gene that was characterized someplace else, it doesn't really tell you what function is. But what we were able to do, my postdoc Oded Beja at the time, was to take this gene and express it in a lab rat, E. coli, so that we could look at the protein function. And without going through the whole long story, what I can tell you is that that opsin is similar to an opsin that's found in Archaea that can generate energy from light in microorganisms. And what we were able to show is that this new opsin, the first one to be found in bacteria, also was capable of generating energy in cells. And in fact, we now know we can take this gene and about 6 others and put it in an organism that can't use light energy and convert it into an organism that can make biochemical energy from light. So that was exciting, and it led to this sort of model of how we think these opsins are functioning in marine bacteria out in the environment. And at first, Marion's right, we were really lucky to run into this. On another perspective, we weren't, because it turns out that about greater than 50% of surface ocean bacteria actually contain these novel opsins. And so this turns out to be a very prevalent feature in microbes that live in surface oceans that can get a little extra boost from converting light energy into biochemical energy. They're kind of like hybrid cars that burn hydrocarbons, and that's what these heterotrophs do. They eat organic carbon, but they also can get a boost from light using these opsins. So that was our first sort of surprise and suggestion that we might be able to learn something about natural microbial populations using these sorts of techniques. So that was sort of my introduction, and now we'll dive into a couple sort of current and ongoing research projects that we have going on now. And the first one I'll talk about is sort of looking at the collective genes from a microbial community to understand something about their ecology in relationship to the environment and how those are interacting. Before we do that, since I put microbiome in my title, I felt like, well, I better define microbiome. So what is a microbiome? It's not very complicated, it turns out. Microbiome is simply a community of microorganisms, or some folks actually use it as the collective genomes from a community of microorganisms. So it's a popular word these days, but I'm not sure why we don't say microbial community. It's probably just easier to tweet microbiome than it is microbial community. So let me begin talking about the study that we have been doing at Station Aloha, which is 100 kilometers north of the island of Oahu. And this site is a long-term study site that a colleague of mine who's here in the audience, David Carl, started studying quite some time ago. And in 1988, he established a research station. And what they do is they steam to Station Aloha once every month, take a lot of samples, biogeochemical samples, physical samples, oceanographic measurements. So we know a lot about Station Aloha over the course of the past 30 years now. It's been an amazing effort, and we believe that time series are really important. One of the things about Station Aloha is that you can get out to the open ocean very quickly, so it's easily accessible, and yet it's representative of a very large swath of the open ocean on our planet. So it's a very large area of the ocean. And one of the sort of hallmarks of what's been done at Station Aloha has been the pH curve, because as we know from industrial processes, CO2 is being added to the atmosphere, and we see the CO2 going up in the atmosphere every year. This is the Keeling curve. And with measurements at Station Aloha ongoing for 30 years, we can see the corresponding absorption of that CO2 and decrease in pH over the course of these years. So it's been an important element in understanding how our oceans are changing in response to human processes. So let's talk just a tiny bit about the environment around Station Aloha, because this will become important in this gene ecology study that I'll tell you about. So one of the things that I mentioned was really important for plants is nitrate. It's like the fertilizer that you put on plants. So they need it to grow. And why nitrate is so low in concentration here is because it's all tied up in the phytoplankton or the photosynthetic microorganisms. So any nitrite that might sneak its way up into the upper water column where there's lots of light immediately gets consumed by the phytoplankton that are there. So these sort of profiles of very low nitrate are very characteristic of Station Aloha. And of course, as light decreases and there's less and less light available for photosynthesis, then there's less consumption of that nitrate and it starts to rise in concentration. And that actually happens right near where this chlorophyll max is. Chlorophyll is really an antenna for photosynthetic microorganisms, and as light gets lower and lower, they basically synthesize more and more antennas to try and absorb that light. So this is a very characteristic feature, and it will become important in our discussion in just a few minutes. So if you can, try and remember these couple things. There's this chlorophyll max that occurs right around 125 meters, which is right where the organisms are running out of light to be phototrophs. And then you also see this increase in nitrate corresponding to this deep chlorophyll maximum. All right, so that's the background, and what we did to study this was similar to those techniques that I told you about, which was to create a gene catalog of the microbiomes or the microbial communities at these sites. And I won't go through all the details, but the point is that we could collect the genes over the course of 2 years in profiles where we sampled at various depths along the water column. And by doing so, we could get an idea of what the distribution of genes was as a function of depth and what patterns we might see in terms of gene variability with respect to these environmental parameters. So that's what we're going to look at next. And what I'm showing you here is clustering, and you can cluster the collective genes that are present at any given depth and look at the similarities from time point to time point. So each of these nodes is a different time point, and the color represents the depth from which they came. And so not surprisingly, over the course of 2 years, the genes from similar depths clustered together for the most part. And then we got some very discrete clusters here at 125 and 200 meters and then lower. And what I'm going to tell you about is that this region here, it turns out, is very special because the nature of the genes and their composition changes dramatically as you cross from this area that's in the photic zone through this deep chlorophyll max and just a little bit below it down to about 200 meters. And so really that's what I'm going to tell you about. And I'll try and define all these terms, but a lot of different core genomic properties, it turns out, change across all the species and the whole community as you descend in depth in these environments. And that includes these elements here, which I'll define in a moment. So let's take a look at that. First of all, let's talk about GC content. And I apologize, I know many of you here know what this is, but I figured I'd cast it general. And so we all know that DNA forms a double helix, and in that double helix are the bases, adenine, thymine, guanine, and cytosine. And these bases are what form these hydrogen bonds that hold the two strands of DNA together in the double helix. And it turns out, and these are known as Chargaff's rules, guanine is always equal to cytosine because those two base pair together in the double helix, and adenine the same, binds to thymine, base pairs with thymine, and is equal to the adenine is equal to the amount of thymine. So it turns out that GC content or AT content is characteristic of DNA. And in fact, different species have different GC contents. And that becomes important in a second. Because what we saw when we looked at the collective genes that we found at each of these depths, so each of these dots is all the genes associated with one sample collected at one time. And each dot contains about 470,000 unique genes per sample. If we just looked at something very simple, like the GC content, we saw a really dramatic change as we drilled down through the water column. So up at 25 and 75 meters, we had a rather low GC content in the collective genes of the whole community of about 32. As we crossed that deep chlorophyll maximum there, the GC content started to go up and reached essentially a maximum once we got down to 200 meters. And at first blush, we thought this was really curious because there was no reason to expect really that all organisms in every sample would behave collectively quite similarly with respect to this fundamental property of the GC content of their DNA. And so what we're going to do is explore that in a little more detail and try and see if we can come up with a hypothesis for why this might be. First of all, we saw this for the collective genes amongst the community. There are ways phylogenetically to just get genes from a particular organism. So we wanted to ask, do all the species show this pattern? And indeed, most of the species show at least a fairly similar pattern as you cross from the shallow zone here in the photic zone into the deep zone. So in general, an increase in GC content, although I'll note the magnitude of that varies in different groups. So for instance, this very abundant bacterium here only increases by maybe 1% or so in GC content, and yet it's quite reproducible across the samples. And this sort of phenomena was actually known from cultures because Penny Chisholm had for very earlier shown that isolates of Prochlorococcus, if you looked at their genome, this is an abundant photoautotroph in the ocean. Ones from the surface had a very low GC, and then ones that were isolated deeper had a very much higher GC. So this is still kind of puzzling because why would we expect microbes to shift with respect to this very fundamental property in their DNA? So what we're going to have to do is back up a little bit and think about this in terms of the properties of DNA and what this might mean. So first of all, in order to explain this, I'm going to tell you a little bit about the nitrogen economy of cellular building blocks, such as DNA and protein. And one thing that we know is that an AT base pair have one less nitrogen than do GC base pairs. So that's interesting because nitrogen is very limiting at the surface, and so the organisms are sort of sparing themselves the nitrogen they might need for a higher GC content there. The other thing that we'll see might be related to this in a second is that the amino acids, the building blocks of proteins, have different side chains in different amino acids. And some contain quite a bit of nitrogen, and other side chains don't contain much at all. So might these things be related to this change in GC content that we're seeing in the water column? Let's ask that question, but my advisor told me when I was a graduate student that 50 minutes in the library will save you 2 weeks in the laboratory. And so if we looked at the literature, there's some evidence that increasing GC content actually is related to the increasing nitrogen to carbon ratio in DNA, so that makes sense, but that actually is also related to the nitrogen content in proteins. And so we can start to take a look at the collective predicted nitrogen content of proteins in these samples to see if there's really a potentially nitrogen-sparing effect amongst these communities. The other thing I'll mention is that models have also suggested that the GC content in DNA can drive codon usage, or the types of code and amino acids that you might use, and I'll explain that a little bit further in a second. So what do I mean by codon usage? So there's redundancy in the genetic code. So the genetic code is triplet, and these triplet nucleotides code for an amino acid. So this is all the same amino acid leucine, but you can have different triplets that code for that. And as you can see here, some of this triplet code has a lot more GC than others, which has a lot less. And this is going to be important in a second part of this model, because what we did was then to look at codon usage as a function of depth in the collective genes in these populations. And as predicted, this is ranked towards the lowest GC types of codons, and those were the most abundant up at the surface. And then as we went deeper, there's a lot higher GC codon usage. Similarly, up at the surface, there's a lot fewer codons available because the GC content is lower, and then that jumps up too. So how does this relate to the protein content? Is it possible that the GC content change that we see in the DNA has an effect on the nitrogen that's in the proteins? And so we took a look at that, and indeed that is the case. If you look at this value up here, this is the number of nitrogen atoms in residue side chains of proteins. And all that means is it's the amount of nitrogen that's contained in a particular peptide. And if we plot the GC content that we saw change as a function of depth, remember, we see an increase in the nitrogen content of the collective proteins in these communities as a function of depth, basically, and across this genomic transition zone. So this is sort of interesting and then leads to a hypothesis about what's going on. We think that in the environment where nitrogen is very limiting, that organisms have basically evolved to have low GC contents. They need less DNA, less nitrogen in their DNA, and they also have peptides which are much less nitrogen containing. And in fact, we can look at that, and if we do that and plot this amount of nitrogen in the collective proteins in the environment relative to a number of different environmental parameters, what we see is there is indeed a positive correlation between the nitrogen you find in these proteins and the amount of available nitrogen out in the environment. And GC tracks that quite well. So as you go deeper, there's more nitrogen, the GC is higher, and there's more nitrogen in the collective peptides. So the reason this is interesting is that people have been debating for quite some time as to why organisms might have different GC contents. And what we think is that it's really nitrogen limitation that's driving this push towards low GC and low nitrogen containing proteins in the collective community and the individual organisms that live there. There's a couple other properties that change in genomes as a function of depth at station Aloha. Yes? Yes, that's a great question, actually. I have to qualify this. These are all prokaryotes, bacteria and archaea. Good question, thanks. So what happens also is that genome size of the average microbe at each of these depths changes pretty dramatically in the same place. So across this 125-meter deep chlorophyll maximum down to 200 meters, we see an increase, almost a doubling of the genome size. And of course, that's going to add a higher nitrogen demand to the organism too as they get bigger genomes. So let's wrap this up and see what we think about this at the moment. Well, the current hypothesis is that up at the surface, where nitrogen is limiting, organisms have this low GC content and proteins that have a low nitrogen content. And if you do the numbers, basically the smaller genomes can save you about 40% of the nitrogen that you get in savings by having small genomes with low GC. And the low GC saves you this much. And then the nitrogen requirement for proteins is a little tougher to estimate because that depends on how much of any protein is expressed at any given time, but maybe somewhere around 10% or so. So what we can say about this is that it's consistent with trends that have been seen from nitrogen-rich coastal environments to the open ocean. So it's kind of consistent with transects as opposed to profiles in the water column. It's also consistent with global ocean survey data from many other different places, so not just Station Aloha. And what we think is that this may be relevant for understanding genome evolution in a lot of different habitats. And we have some ideas and actually some data about that I can't talk about, but we're excited about this because we think that understanding the relationships to even basic properties like this of genomes in relationship to the environment can teach us a lot about how microbes are evolving. So I'm going to transition from that story of the ecology of genes to microbial dynamics. And that's a challenging topic because we have to think across many different scales. If you're an oceanographer, you think about all sorts of processes, small turbulent patches or plankton migration through hundreds of meters Turns out some plankton come from very deep and go to the surface every night to feed and then go back down. Phytoplankton blooms fronts and eddies on all these different scales from meters to kilometers and from minutes to months. From a microbial perspective, you have to think about different scales as well as these oceanographic ones that have to do with what the world looks like from the perspective of a single cell to what populations are actually experiencing and what population averages really mean, up to the level of communities and so on over time periods of maybe seconds to years and very different sort of volumetric scales. So we can't study everything all at once, and the scale that I'll be talking about in a little bit really is right around here, the scale of what happens over the courses of hours to days and what happens, say, in a teaspoon of seawater up to maybe a liter or so. And one of the things that we decided to focus on, because there's so little known about it, and there's evidence that suggests that daily variability in microbial populations might be very important for processing of materials, photosynthesis, and so on, was to look at diel cycles. And we do know quite a bit about circadian rhythms. Of course, we have circadian rhythms, as do many other animals, fungi even, and cyanobacteria. So from model systems, we know quite a bit about the molecular biology of molecular clocks that maintain these diel rhythms. But if you take a look at the diversity of bacterial life, we don't know a lot at all. So here are the organisms we know about in terms of diel cycles, and here's the rest of the phylogenetic tree. So it's a pretty open playing field in terms of how diel cycles might be affecting organisms out in the environment and whether they're important to ecological processes. And so what we're doing is diving again into, in this case, the central dogma of molecular biology and looking at the informational macromolecules that can teach us some things about what's going on out in the environment. And in particular, we'll focus on transcription, which is really the expression of genes, which is a very dynamic process. Genes can be expressed very quickly and then turned off very quickly. What we've been able to do over the past decade or so is use studies of gene expression, not just in the laboratory for a single model organism, but for collective groups of organisms. Again, sort of using Pace's logic, where we reach in and we grab these collective macromolecules from the whole community and isolate those. And then we look at the patterns of RNAs as they come and go over short periods of time over the course of the day. But it's not that simple because you want to follow a population over time at basically consistent sampling, and that's really hard to do for microbial communities. Imagine if I was on a mooring and I put my sampler here, or I sampled in one place, we'd really be sampling the sort of superhighway of currents that we're passing by that might or might not be the same population of microorganisms. So what we do instead is Lagrangian sampling, and Lagrangian sampling is just riding with the microorganisms in the same fluid environment so that we have a better chance of sampling the same population over time. And the way we did that initially was working with a colleague of mine at the Monterey Bay Aquarium Research Institute. He had developed a robotic sampler. It's an electromechanical fluidic device that can essentially float in seawater, take a sample of seawater, put it on a filter, and then preserve that so that we can then go back and do these molecular studies. So in a way, this is sort of our camera. It's taking pictures over time by collecting those microorganisms. And then we develop the film to look at the sort of instantaneous gene expression across the community at that point in time. So and this is just a picture of the device that's now floating on a big float so that it can take these samples over time, say every 2 hours in a Lagrangian fashion. So we're kind of taking a movie, if you want to think of it that way, of these microorganisms over time. So I'm going to tell a story about, very simple, I'm not going to show you too much data, but I'll show you a little data from some of our first results. But we, of course, expected to see diel rhythms in the phototrophs because that's well known and it's been very well studied in the laboratory. And in fact, if we compared the diel rhythms of the phototrophs out in the environment to what's been seen in laboratories, the match is pretty incredible. There's very good sort of reproducibility of peak time of expression for each gene in the organism. But the big surprise was that these organisms that are eating organic matter and aren't using sunlight energy very much to get energy were also diel. And in fact, they showed a little bit staggered patterns. So these are transcripts for respiration from 3 different types of heterotrophic microorganisms that also show a pretty reproducible diel rhythm. And you can see that in a lot of these different organisms. And so that was sort of a surprise because it wasn't necessarily expected. In fact, I'm not sure yet that this has been demonstrated for free-living organisms in the laboratory. So it was a bit of a surprise. All these organisms seem to be really keyed in terms of their gene transcription, their gene expression patterns to the course of events over the day. So now this is a little bit of a messy slide, but I want to walk you through the reasoning here in terms of understanding how transcription of many different genes across many different species may tell us something potentially. We think they may be telling us something about their potential interactions in the environment. So what this is a heat map. Each of these blocks is a day that was taken by this device. So this is a time point here in this column. And each row is a collection of genes that behave similarly in time from a particular organism. So as you go across the row, you'll see it turn red. That means the genes are being highly expressed. And then it goes back down again. And then we can see that for many of these clusters of genes from a particular organism, they behave the same each day with these rhythmic patterns over time. And so what we aim to do was to try and look at those expression patterns between many different microorganisms. So what do the collective genes that behave the same way in time look like when they're from different microorganisms? And can that tell us anything about their functional attributes or what might be going on in the environment? So let's take a look at one of those clusters. This dendrogram here is just clustering by similarity of behavior in time of these collections of genes from these different microorganisms. This green is from a photoautotroph, and these other colored ones are from heterotrophs that are eating organic carbon. So these are all genes that are, I'd call them evening genes, that are lighting off at around in the afternoon towards the early evening every day in all these different microorganisms. So how about if we drill into those genes that are behaving similarly in time in terms of expression and ask what their functions are and if that tells us anything? And I don't expect you to read any of this. It's quite a mess and very small print, but I do want to make a point, and that is this. If you look at particular clusters of those genes that are clustering together and behaving similarly in time, they tend to have similar functional attributes. So for instance, some of these genes that have particular time-resolved attributes are firing off, like those for glycine metabolism, at very similar times of day. And even sort of what I'd call boutique sort of metabolisms, carbon monoxide metabolism, which only few organisms can do, even the different species seem to be turning their genes on at the same time. And what we think this may be telling us is that out in the environment, different metabolites may be rising and falling over the time course of the day, and that's inducing some of this gene transcription in response to that, and may be indicating transfer of materials between these microorganisms in these mixed populations. And so we've used a couple of different techniques to apply this approach, but one of the more surprising things is that if you conduct similar experiments in both a coastal region, which is very different, very cold waters, very high nutrient, very shallow chlorophyll maximum, compared to the open ocean, we see quite similar patterns in some organisms, and that's going to be more apparent in this plot. And what you see here is just a list of different organisms whose genes we've looked at in terms of how they're expressed, and those are plotted here as to whether they're diel or not. So if you see a solid color, those are genes that are turning on and off very regularly over time, and from all these different microorganisms. And what you can see is a pattern over particular pathways. This would be for protein synthesis. And this organism's sort of firing off first, followed by this organism and this organism over the course of the day. And that's for the coastal region. We have similar sorts of organisms in the open ocean region, and one of the more surprising things to us is if we looked at protein synthesis genes and when they're expressed, we see a similar order of expression patterns when we look between the coastal and the open ocean. And so what we wonder is if this really is indicating metabolite exchange between these organisms over the course of the day, is whether that's really conserved even amongst organisms in very different habitats. That remains an open question, but the regularity of the patterns here is amazing. I mean, I don't know how to describe it to you very well, but we're just looking at seawater. This is just seawater with microbes in it, and yet we're seeing these very predictable and regular patterns in terms of their gene expression. So here's what we think may be going on, or the questions that we're asking, is that does trophic transfer of materials, say from a photoautotroph, which is making organic material, in a diel pattern, cross-feed other microorganisms such that they entrain, and they also fire off their genes in sort of a staggered but sequential pattern that leads to a lot of regularity. So we're thinking about those questions, and with our colleagues, trying to understand if this may be true, using a number of different types of experiments that I'll talk about in a second. And what our hypothesis is that this transfer of materials could potentially coordinate the activities in metabolic cycling between these different groups of microorganisms. Now, this is data pretty hot off the press from one of my colleagues in SCOPE, actually Angie Boyson and Anitra Ingalls, and what they've been doing is metabolomics and looking at metabolites that are produced in cells over the course of time using very similar sampling approaches. So they'll filter a sample over time, and then they'll look at those samples with respect to metabolites using metabolomics, and what they see are regular diel cycles in many of the small metabolites that are produced in cells. And so what we're attempting to do now, and I would have to say it's early days, is to see if this metabolite expression by some of the producers, if you will, corresponds to transcripts or gene expression for utilization of these compounds, thereby potentially entraining the sorts of patterns we're seeing. It's not an easy thing to do. I have to emphasize again, we're talking about raw seawater, and we're actually trying to do these very precise techniques with metabolomics and transcriptomics, but the early results are actually encouraging and we think quite promising, and we're hoping in the context of the SCOPE project that we're going to make progress in this area. And so I've got about 10 minutes, and so I'm going to finish up with a recent collaboration amongst many people in our project in 2015, where we conducted very similar studies over the course of time, and we can let the ocean help us in doing these studies, because there are oceanographic features that actually entrain water, and these are called eddies, and there are some that are counterclockwise and some that are counterclockwise, and they sort of trap bodies of water that makes our Lagrangian sampling actually quite a lot easier. So we went on an expedition to do some of these studies and looked at our colleagues together collectively, looked at a lot of different parameters in terms of trying to understand the daily dynamics, the day in the life of these small microorganisms. And I'm just going to show you a couple anecdotes from that study, but one thing that we're pretty intrigued by and a little bit excited about is the fact that some gene expression can actually potentially be predictive of cellular processes. So in this top panel here, what you're looking at are transcripts for a gene that's called CEPF, that is one of the proteins that's involved right before the cell's going to separate. So cell division protein. And what you see here in this jagged line, and this is data of Ginger Armbrust and Francois Riboulet, is that you can see the cell division process in this very same organism, where you see the transcripts firing off right before cell division. We see things for other important cellular processes in this organism, such as nitrogen fixation. So here are the transcripts for nitrogen fixation in these solid peaks, and then here's the actual process of nitrogen fixation measured. And so what we can say about this is that at least for some processes, not all for sure, that the transcripts may be predictive of at least the timing of these processes, and that way may be able to serve as a proxy for some processes that organism specific that are otherwise really hard to measure. And so we're pretty encouraged by this result. You can look at another protein, this one called FTSZ. We can look at the gene expression of that amongst two organisms that we know divide at different times of day. So here's a photoautotrope Pleurococcus, and here's its cell division FTSZ protein firing off late in the day here. And we actually know that Prochlorococcus divides right in the evening, right around 6 p.m. If you look at that the same transcripts for that protein in another cyanobacterium, Crocosphaera, what you'll notice is those are reaching a peak towards the middle of the night or so, and we know that Crocosphaera actually divides out here at the beginning of the day. So in some sense, looking at these expression of these cell division proteins may be a proxy for us to look at organisms whose cell division processes aren't even known. And that's one of the avenues that we're taking in the project. And then I'm going to finish up by talking about the more abundant entities in the ocean than even cells, and those are the viruses. And there's been a lot of really intriguing work on what I'll call wild viruses in the ocean, and they've been thought to be quite important in a number of processes. Because they can lyse phytoplankton cells, they can sort of short-circuit the classical food chain and add more dissolved organic material. So they can have effects on nutrient cycling. Interestingly, these viruses also can carry the genes of their host and move them around. And so they're also important in terms of gene transfer and also in generating genetic diversity as cells try and develop evasive mechanisms to the phage, and the phage evolve ways to get around that. So they may be important, actually quite important, for generating diversity and robustness in natural populations of organisms. So what we were able to do from this Scope 2015 study was actually assemble either whole phage or fragments of phage genomes and look at those individual phage over the course of time. So each of these rows here is one individual genome from a different virus, and I'm not going to dwell on this too long, but one thing we really didn't know was what the sort of persistence of viruses out in nature really is. So over the course of days, we can see that these individual genotypes are really quite persistent and very abundant, it turns out. And even over the course of over a year, a number of these viruses appear to be very prevalent over the whole time. And with colleagues in our project, what we're hoping to do is use genetic data from the hosts and from the viruses to start to back out the network of interactions between host and viral species. And then finally, what we saw was that just like their hosts, it looks like the viruses have daily cycles, diel cycles. And so what you can see here are the viral transcripts collected from the whole community. And what these data are indicating is that the viruses are actually piggybacking on their hosts and having sort of daily infective cycles as opposed to random ones. And we're still trying to figure out what that means and how that might help us associate viruses with their hosts. But we think that it may be very important in terms of how these processes occur out in the environment. So I've got 5 minutes, and I guess I will finish up just with a little bit of thinking about where it looks like the field is headed next in terms of looking at microbial communities out in the environment, and particularly in the ocean. And what I've tried to make the case is that we can actually use genetic information, both from genomes and genes, to make inferences about what may be going on with community metabolism and evolution, potentially even in host-parasite interactions and towards ecosystem function. There's a lot more examples of that from other laboratories that I didn't have time to talk about today. One of the things that we think in terms of being able to get at this space-time continuum, if you will, and higher resolution, it's clear that the ocean's undersampled on a variety of scales, but the use of some of these new technologies to get higher frequency sampling is something that offers an opportunity to really delve a little bit deeper and trust our data a little more. And so we've been working with the Monterey Bay Aquarium Institute to develop these robotic sampling vehicles that can actually autonomously sense features in the ocean, like this chlorophyll max here, and then make a decision once they've determined where the chlorophyll max is to go ahead and drift along and sample that, and then go out and look for another feature. So these are sort of exciting capabilities that we'd like to think about to have a better sense of when an event happens out in the ocean, that we can get to it and sample it and better understand it. And then finally, besides better observations, we also need better modeling, and I've borrowed this from my colleague Mick Follows, who is also in the SCOPE project, and he's started another project as well called Sea Biomes. And we need to be able to use some of this high-resolution data and interact with the modeling efforts so that we can better predict what's going on in the environment now and where it may be headed. And so I think I'll finish up there with questions. Now I'm going to get a little poetic, so I hope you'll forgive me. But I've tried to make the case that over the course of Earth's history, I mean, if you think about it, our climate and our redox state, that is our atmosphere, the way our oceans behave and absorb energy and light, as well as our terrestrial systems, are a direct result of the evolution of microbial species. So over 3.8 billion years of evolution, microbes have shaped our Earth, but that's in the face of the past, say, 150 years and the evolution of this species, which now has a greater impact on some biogeochemical cycles than our 3.8 billion year old friends. So for instance, we now fix nitrogen by the Haber-Bosch process, more nitrogen than all the microbes combined, and yet 200 years ago, they were the only source of fixed nitrogen. So things are changing, and I think we have actually responsibility to try and understand how the environment works and how it's changing and what it will be like for our children and their children. And so I think I'll finish up with that. I'd like to thank the people who did all the work in my lab, as well as all the SCOPE collaborators, those who I've mentioned and those who I haven't, and of course all the generous funding that we've had to help support this work and try and push the envelope on understanding the microbial ocean. And so I think I'll finish up there, and I'm very happy to take any questions.